Hi, this video is actually about continuations of the first part of biological fluorophore. So we have came to know that what is the biological fluorophore, what is the incident of fluorescence, and then we have came to know that what is the Jablonski diagram, isn't? So Jablonski diagram is a diagram which can show you this fluorescence phenomena, and as we have came to know that there is a three-step process in the whole fluorescence. The step 1, step 2, step 3. Step 1 is the excitation, and step 2 is the relaxation, and step 3 is the de-excitation. So here you can see in this image, ground state is S0 and excited state are S1. The S1 is the first excited state, and then you can consider second excited state, third excited state. Okay, so they are all the excited states, first, second, third. And as we said, fluorescence phenomena is like that. Whatever be the excited state, first, second, third, fourth, whatever be the excited state, you have to first come back. You have to first come back to the first excited state, means S1, and then only you can return back to the ground excited state, a ground state, and that is called S0. So whatever be your excited state, you have to come back for the excited state S1, and the first excited state, and that is called the relaxations, and that has been shown by the curly arrow in white color. That has been shown the relaxations. Okay, and that's why for this relaxation actually the molecule already are losing some energy, and that's why the energy of the emitted light, this one, energy of this one is the less than of this one. So as you can see here also, energy is decreasing. That's why wavelength will increase, because wavelength is inversely proportional to the energy. That's why you can see here you are applying blue light and you are returning back with a green light. So fluorescence is that's why is very much advantageous for bioimaging, because you are applying a low wavelength light and you are getting a high wavelength light. And high wavelength light always used for signaling. For red signal you have seen, and orange signal, because that are attractive. So that's why in your image you will get a very attractive look, a very lighty look, which is very useful to investigate the very small details of your cellular system. So that's why it's very advantageous, and that's why people use the fluorescence phenomena very much. Okay, so that's a real advantage, and that the reason you have came to know that what is the reason. This is the relaxation. What is the reason? The relaxation. Due to this relaxation, actually the emission wavelength is actually greater in wavelength as compared to the excitation wavelength. Okay, now what we will do, we will figure out in a short video the whole concept all we have learned. Okay, so we will see a very short video which will say what is the fluorescence phenomena and why it is used in biology. So one thing again, I forgot to mention that fluorescence is actually means fluorophore which shows the fluorescence is actually going to be tagged. What happens? Fluorophore has some chromophore, means chromophoric region, that can accept energy and can return back to some another low energy, like this fluorescence. Okay, so whatever fluorophore you see, like DAPI or ethylene bromide, you will see that are very well known fluorophore for biological experiments. And what they do, they first take the light and leave and release some another higher wavelength light. And this type of fluorophore actually a very long pi moiety have. So that's and that type of organic molecule also have some functional group like OH and H2, and that's why they can be conjugated with any organelle you have present in your cell or tissue. That's why it's very useful for imaging purpose. So what happens, let's say I want to image a nucleus. I want to image a nucleus of a cell. So what I will do, I will take the cell, okay, and incubate, and for first culture in the cell culture flex, and then incubate that cell with DAPI. DAPI is a very well known organic fluorophore to stain or to tag nucleus. So what we will do, you will incubate DAPI for let's say 1 hour, or 1 hour also is not required, 5 or 6 minutes it already tag. So you will incubate the DAPI with the cell, and then DAPI molecule is goes inside the cell, and DAPI has means every fluorophore actually has some selectivity, so that will only target nucleus, only bind to the nucleus. And whenever you are giving light, so only DAPI will be excited and emit some light. Okay, and then when you will take the image, okay, when you will take the image, this emitted light actually going to be recorded by some detector and it will be converted to some image. So in image you will see only the green light which is coming from the DAPI. The emitted light of DAPI is actually green, blue. That's why blue color we will see for DAPI, and that's why you can say that blue coloration is for the nucleus. And whatever the inner phenomena happening in that is the fluorescence phenomena. Let's see. Okay, so here you can see this is also a stained cell where you can see this green colors, some plasma membrane or something like that. So this all happens due to fluorescence. This also some staining has been happened due to some fluorescence, and this are neuron you can understand, and this cell division you can see also. Okay, the nucleus are just dividing, whole cell are dividing, and this also due to fluorescence. Now here you can see now you will see some fluorophore which is very well known. This is DAPI. So DAPI, look, that you have some NH2 group or double bond NH, and this looks like DAPI looks like very blue, intense blue. You have ethenium bromide and GFP also. It's a protein. GFP protein actually can show fluorescence. And now look at the phenomena of fluorescence at atomic level or molecular level. This is the electronic energy level, this is the ground state, and this is the excited state. When electron will be excited on absorbing light, and then it will create relaxations and come back to the first excited state, and then it will come back to the ground state, first excited state to ground state. Now look very carefully, this is blue, means the emissions light is actually higher in wavelength as compared to the excitation light. So emissions light will be always higher in wavelength in excitation. Now let us take the example for microscopy, how it is used, the fluorescence phenomena. So here you can see the laser source, it will come, here are some mirror, it will come here the specimen, here is the specimen, and it will make some fluorescence phenomena, it will return back some emission light, it will go to the mirror and then come to the eyepiece. Now you can see the image. That's all about fluorescence. So fluorescence is a very important technique, important phenomena which is used in the bioimaging. Hope you understood all the concept. If you have any questions, you just can mail me in the following mail I have given in the description box, and you can also make comment on my video. If you have any questions, I will happy to answer. Thank you for watching. Bye bye.